Alright, hi year 11, this is Mr. Lim here again, and this is our last video on chromatography. We're just going to compare over chromatography types. Okay, so let's get started. We're going to find out what are some advantages and disadvantages of each type. Alright, so first of all, we've gone through the thin layer chromatographies, which is your paper and thin layer. Alright, so what this is used for? So polar and water soluble substances. Typical samples, or like, drug detection or dyes and food stuff. Okay, so the idea is that this is super cheap because it was one of the first ones used. It's basic lab equipment needed, and it's easy to perform. However, you don't get much precision and accuracy from it. So if you remember what happens in a thin layer chromatography, you're going to get these kind of splodges, right? You're not going to get very neat bands. So if you're trying to find things that overlap each other, that's going to be difficult to do. All right, if you wanted this to be better, you would actually get something that would look more like this, where you've got very, very high precision, because you can see that, okay, that only occurs at a small period of time, and therefore that RF there is quite precise compared to an RF of something that's there where you're like, oh, where's the center of it? It's kind of like there-ish, and you're like, hard to tell what that is. All right, so other things about paper and thin layer chromatography is that it is samples need to be colored or visible under UV light, otherwise you need to use some sort of staining, which is not ideal. But yeah, that's the idea. It's nice and simple, and it was our first type of chromatography developed, but not very good, not compared to the new types. All right, so then we get to our high performance liquid chromatography, right? And what you'll notice is that for high performance and gas chromatography, these are effectively the same, all right? So it is definitely more expensive. You have a much higher precision. You'd need a smaller sample size. Okay, so that means you need less of the sample to do anything about it, and it's readily automated, which means it can be done by a computer, right? But the differences between these two, right, are, okay, the HPLC, you need a medium to high molecular mass organic compounds, so things that are kind of larger, all right, foods, drugs, biological samples. And these substances must be soluble in a suitable solvent. So you must be able to dissolve them in some sort of liquid mobile phase. All right. Compared to gas chromatography, well, now you can do low molecular mass organic compounds, which are much smaller molecules, right? Like acetone or aspirin and things like that. So you're testing water samples, gas samples, foods, drugs, and biological samples as well. However, the problem is that these things have to be vaporized, which is why they have to be so small and a lot more stable. These high and medium molecular mass organic compounds will tend to decompose at higher temperatures. And so when you try and vaporize them, they'll break apart, and therefore you're not going to be able to work out what they are. All right, so it just means that these things can only be done with smaller things, which are a little bit more stable and can vaporize because they have much lower dispersion forces, and they are able to turn into a gas much easier compared to the higher sized compounds with greater dispersion forces. All right, so those are your different types of chromatographies, right? That's what you need to kind of remember, the similarities and differences between them, and you just generally need to be able to talk about what are some advantages of HPLC or GC against thin layer or paper chromatography, or what are some differences between HPLC and gas chromatography, that kind of stuff. All right, that's it. Adios.